This is what the title is referring to. We're talking about like biological systems are composed of multiple entities and objects, or heterogeneous types and interrelated. So that's the first part of the title. The second part is basically it's mentions basically what we are going to kind of do with all this data as part of this project that I'm going to describe today, which is called the Biozon project. Okay. So I'd like to start with, just as like a point of reference, I'd like to start with the Human Genome Project. It's been already 10 years since this project has been completed. And when it was completed, when the Human Genome was completed, it was completely sequenced. It caused a lot of excitement, naturally. But this was actually just the first step. What this project accomplished was basically determining the 3 billion nucleotides that make up our genome. And that was a significant achievement, but it was actually just the first step towards understanding our genome, our code, our own code, on multiple levels. And because when you look at the genome, again, I'm kind of like, whatever I'm talking about now, assume we already were exposed earlier today, so it's just kind of a certain point of view. So when you look at the whole genome, you can think about it as a master code. It codes for everything that is going on in our cells and tissues. So we're talking about the human genome. And so we have the DNA, the whole, to chop it, all those different chromosomes, but altogether we have those 3 billion nucleotides that are the master code for the cell. And it codes for multiple entities on multiple levels. So at the very basic level, it codes for proteins and RNA molecules. So these can be kind of perceived as the basic procedures in computer science lingo, if you kind of make the analogy with computer programs. And so what we have here is basically each of those entities, each protein or each mRNA, each RNA molecule that has a certain function is basically a basic functional procedure that takes care of a specific of something specific in the cell. So at the next level, we have those basic procedures: proteins, RNA molecules, and also the DNA itself can interact with each other and form complexes or functional modules. We can view those interactions as functional modules that, again, perform a certain kind of like function, really more complex than the function of the individual entities. Often it's those interactions are the mechanism to store and communicate information between different entities, between different molecules. At the next level, we have those entities, those functional modules, together with the basic functional procedures, the proteins and DNA, they are put together in even more complex constructs called cellular pathways. So those are complex cellular procedures, the counterpart of basically computer programs. And they have a specific input, a specific output, those the input and output, basically molecular input and output. And this is just this kind of a simplified view comes just to kind of like gives a glimpse of kind of like how basically the complexity of what's going on in our cells. Again, going back from to the master code, and there's this pipeline, basically, of information, starting from the basic elements through the functional modules and through the more complex cellular pathways. And this, when you think, when you kind of look at all of it together, so what's kind of the analogy that comes into mind? What's the kind of like? How would you describe everything that is going on in a cell? Again, going back to the computer science lingo. So it's like an operating system. Right? You have this kind of very, very complex operating system. It consists of many different programs running in parallel. And those are the computer programs, they are the counterparts of the cellular pathways. But there is, they also communicate with each other. All those different pathways, they have crosstalk. So, and they, we're talking about like an operating system that works on elements. And it is obviously very complex, and many of the elements that are part of this system, we don't really know what they do, we don't really know actually even what they are. So that's, many of those elements are unknown. We don't, although decades of research has already accumulated a lot of information and knowledge about what's going on in cells and tissues, but probably our knowledge as of today might be only the tip of the iceberg as far as what is still out there, what is still to discover. Now although I was making this kind of analogy between cells and computer programs and operating systems, and this kind of analogy can go only as far as a certain point, because one of the things that do differentiate between cells and computer programs is the fact that the master code itself can change, can be reprogrammed. So it can change as a result of random mutations. So it's kind of a natural evolution of the code itself. Or it can change as a result of interference with external agents, like pathogens, for example. So viruses or bacteria can actually cause changes in the genome and obviously change its function. And I don't know if you ever thought about why obviously computer viruses, the reason the name computer virus was chosen to describe those programs is inspired by what's happening actually in the system when viruses are introduced. So it's in order to kind of characterize all those different entities that make up cells, we're talking about decades of research, intensive research into all those different entities, and the result of all this research is stored in many different databases, containing information about different data types. So we have databases that store information about sequences, like DNA sequences and protein sequences. We have databases that store information about protein structures, databases of protein-protein interactions and other interactions, databases of pathways, and so on and so forth. So, as I said, there's already a lot of information that was gathered about all those different entities, and this is just a short list of the hundreds of biological databases. There's specific. This is a link to this journal. They keep track of. There is a link to almost any biological databases out there. Most of them are very small, but many of them are still large and contain massive data sets about all those different entities. So we have all this knowledge about different entities of cells. And while very useful, all this information has accumulated over the years, the problem is that it also presents a big challenge or a need for data integration. And this is, the reason is because most of those databases are highly focused. So when you're looking at each one of those databases, usually stores information about a very specific data type. Either just protein sequences, or just DNA sequences, or just interactions, just pathways. But in reality, the fact is there is a very strong dependency between different biological entities. When we're talking about, again, going to the first slide, like there is all those functional procedures, proteins and RNA and DNA, and they interact with each other, and they're part of pathways. So in order to really, if you want to characterize the biological function of any entity, you really need to look at all the interrelated entities. You cannot just look at just the protein sequence or just the interaction. You really have to understand all the components. So in other words, you need to kind of characterize the broader biological context of any entity to kind of effectively characterize it. And so, for example, if you want to characterize the biochemical function of a gene, so that function depends on the interactions that this specific gene forms with other genes or other molecules in general, the specific pathways that this gene participates in, its subcellular location, its expression or activity level under different conditions, its subcellular location, and so on. So there's a lot of information you have to take into account, and that's why there's a very strong need to corroborate and integrate data from different resources, from all those different databases, and different aspects of biological systems in order to analyze them effectively. Right? And I mentioned all those different databases, and we're talking about hundreds of them, so they're kind of like kept, they're maintained by different research groups or different centers around the globe. So you have a massive amount of data, but it's you have to kind of, it's kind of like you have many bits and pieces of the big picture that you have to put together in order to really understand what you're looking at. So, now the challenge of data integration is surprisingly difficult. And it seems like, even if you do it on a specific, on a single entity, let's say you want to study a specific gene. So you start by searching information about this gene in different sequence databases, and so you start by just searching those databases, and you have to unify all those the results that you get from those different databases into a non-redundant and consistent set. And along the way, you have to deal with different formats, different databases. They use usually kind of structure, the same structure, kind of formats, but each database has its own format. They have their own system designs. They use different identifiers, meaning kind of session numbers. So they might refer to the same entity, but one might call it like, Tim, the other one might call it Jack. So you have no idea who is who. Those session numbers are completely arbitrary, right? So you have also some databases do maintain links to other databases in order to kind of help with figuring out which entity is which, but those are only very partial, and also usually they are not established with coordination, in coordination with the other databases. So what happens is that often it's out of sync. Let's say one of the databases updates their data, the other one basically, you have the problem of dead links, or or links that point to the wrong entities and so on. And you have also some issues of other inconsistencies. So this is, just thinking about that. You start from one gene sequence, you might end up spending weeks just collecting the information of that one gene sequence. Data integration on large scales don't have this. So this is difficult enough task, difficult enough task just for individual entities, and when it comes to large scale data sets, it's becoming overwhelming. So this is kind of like to summarize, data integration can be a significant task, especially on a large scale. Another complexity that arises because of the nature, because of the fact that data is dispersed among all those different databases, is that it's very difficult to query the data in ways that it utilizes mutual dependency between entities. And later I'll give examples that will make it very concrete about what do we mean by utilizing mutual dependency. So and another big problem with this the existing situation is that branch propagation becomes the bottleneck of many research projects. Just in order to kind of be able to address any research problems, you have to start by collecting all the different data sets, creating this kind of unified, non-redundant set that kind of would just to kickstart your project. But if you already at that point fail because of technical kind of issues, so obviously that's a major kind of problem, and it slows down research in biology and medicine. And this is especially aggravated by the fact that when we look at the pace that biological data accumulates, this is something that is it's pretty overwhelming. So this is kind of here, for example, we look at the and those graphs only go until 2005. But here, for example, we see the number of structures, the protein structures that were determined over the years until 2005. We have this kind of exponential kind of growth in that number. Here we have the number of interactions that were determined. Again, we have this exponential growth of information, of knowledge. This is the number of publications, number of articles a year. So it's again just growing and growing, and the pace keeps increasing. And this is the worst graph of all of them. This is the number of base pairs that have been determined. Again, the graph stops only in 2005, and with new technologies, it just goes kind of faster and faster. This is basically the pace in which new sequences are determined is super exponential, which means it evolves in a pace which is faster than the pace in which computer technology evolves. Which poses a major challenge, additional major challenge, when you have to kind of put all this information together. You're dealing with something that it's another level of complexity you have to deal with. And just as a side note, although I'm not going to talk about that, one of the reasons this kind of whole summer school, you heard many talks about biology, and originally, the core of the center is about information theory, right? And kind of trying to reach out to other disciplines. And one of the reasons this is very relevant, information theory is very relevant to those challenges, is because in order to address the super exponential growth with data, you have to present methods from, for example, information theory, compression, very smart compression algorithms that are tailored to handle biological data in order to address that. Because otherwise it becomes unmanageable. But this was just a side note. And the second side note, you heard, I mean, it might have been mentioned in one of the talks previously, did anybody mention big data? It's kind of this, no? So this is kind of like one of the new buzzwords, the big data. So basically, many resources, the Obama initiative, the Obama administration has announced a new initiative to devoting basically a lot of resources to address big data. And when we talk about big data, it's basically developing technologies and novel methods to address large data sets. Biology is pretty much at the front here as far as the challenges it presents. So just a side note. So this is so this was kind of this was the setup to explain what I'm going to show you kind of today, which is the Biozon system. So the Biozon project was initiated in order to kind of build this unified biological knowledge resource. The goal was to create a working system for storage, retrieval, and exploration of biological data over entities ranging all the way from the molecular level to the cellular level. And this resource integrates multiple databases, we're talking about tens of databases, plus in-house computer-generated data, which is data that is derived from the source data that we got it from other databases. So this is just to give you kind of an idea of like what kind of data we have in this system. We separate data into two general categories. We have source data, which includes information protein sequences. Those are names of databases that, pretty major databases in this field. We include information protein structures and DNA sequences, and protein fractions, and pathways, expression data, gene ontology, and so on. And then in addition, what Biozon does, it puts together all this data, and I'll go back in more detail in the next couple of slides. But in addition to that, it also augments all this data with metadata, which is computer data that we derive from this raw data. So for example, this includes, which focuses on those, like similarity information about basically current similarities between different entities, for example, between proteins and protein families, based on sequence, based on structure, based on other attributes. These are very significant when you explore the data because they help you to properly acknowledge from well-studied entities to uncharacterized entities. So this is just giving you kind of like a hint of what's coming next, but this kind of metadata really enhances the knowledge that is stored within that resource. So there are other types of metadata, I won't go into kind of like all this, but right now what we have in Biozon is about like 100 million documents and 6.5 billion relations between all those different entities. And all this information is stored in a way that is, makes it really possible, like the goal is to create a system to make it possible to utilize those interconnection between entities and leverage on that. Basically being able to kind of explore the context, this kind of broader biological context of entities in order to characterize entities better and come up with better functional predictions, for example, or being able to associate entities with, let's say, diseases or certain disorders better than what you could do by just going into the individual databases. So next I'll just give you kind of an idea of like the main principles. I'm not going to kind of, I said I'm going to keep it light, so after kind of describing the main principles, I'll jump to kind of a demo, just showing in real time how the system works. Okay, but I'm just going to give you an idea of like what are the main principles behind the design of the system. So the first principle is a warehouse approach, which is basically we took the same approach that Google took when archiving the web. If you want to query something efficiently, you have to first create a local copy, because otherwise, it pretty much, that's one of the, if you want to enable advanced data manipulation exploration techniques, you have to go with with this stuff. I mean, these days things are changing. You know, with the first whole cloud computing concept kind of makes it possible to kind of spread the effort. But it's still, you know, the main kind of the core of the system still, you know, like in order to really to be able to answer queries in real time, most efficient way, this is kind of I would say the safest way to go. And it employs what we call vertical integration with tightly connected schema. So the tightly connected schema refers to the fact that all those different entities are highly dependent on each other. Proteins encoded by DNA sequences, proteins can interact with each other, they can be part of pathways and so on. So we have those kind of, all those different entities and all those relations from them. Vertical integration means that when we download the data from the different databases, we do not treat them as complementary. I mean, we don't just put them side by side, but rather we actually try to figure out what are the entities that are identical in the different databases. And basically we try to kind of like, we're dealing with overlapping data sets. Those are not independent, kind of unrelated, but rather they're overlapping. So this kind of, by going with this kind of like route, basically the goal was to try to eliminate redundancy and being able to basically create all those unified, non-redundant sets of entities coming from different sources. And you mentioned provides great control of query execution and optimization, which is being able to answer queries in real time effectively. The second principle, it's kind of refers just to the design of the and the bolts and things of the system, but it's the data model that is kind of in the back end of when representing those entities. We had a combination of the graph model and the class hierarchy of entities and relations. So the graph model seems kind of very natural to describe all those the relations between entities. We have a node for every possible entity, like a protein sequence, DNA sequence, protein structure, and pathways, and so on. And the edges are basically the relations. They kind of determine the relation between them. The hierarchy, the reason to introduce hierarchy is because many entities are related in a kind of formally related in a way that you might want to take advantage of, inheritance properties. That if you design protocols and applications for a certain data type, they could be applicable to another data type. So introducing hierarchy kind of helps you with with building such a system.
Rotating protein structure.
Well, actually, protein structure and protein sequences are not kind of, but DNA sequences and protein sequences are part of the same. They're both sequences.
Okay.
So that's kind of
that kind of.
Yeah, that kind of yeah. So so hierarchy kind of like implies inheritance, which also maintains extensibility. The graph model is more appropriate in describing the complex types of relations between biological entities. And this combined approach, it provides us with more flexibility when we need to describe any biological entity. So when we need to introduce new biological entity, we have the flexibility of either putting more constraints on the way it's connected to other entities or more constraints on the way it's described in the hierarchy. So it might kind of like, this kind of description, I could see how it might be kind of a little bit detached, and, but just to give you an idea that, that it just, there are multiple decisions and design choices when you kind of like start building such a system. This is just one element of many that you have to address. What we see here is just kind of like a tiny subgraph of the whole biological data graph that we have. And so we have those different entities, protein sequences, DNA sequences, protein structures, interactions, protein families, pathways, and as a result of all this integration, what we have at the end is like something that looks like that. For example, we have this DNA sequence encodes for this protein sequence as well as for this protein sequence. It's also a superstring of there's another shorter DNA sequence that is just a fragment of the longer one. This happens a lot. In biological databases, you often have fragments with partial information, and maybe one of those entities is associated with some information, while the other one is not. So kind of being able to transfer the information from one entity to another is very crucial.
So the edges are directed?
No, so all those edges are undirected. You can't. I mean, each one has you have both ways. Kind of like for example, DNA encodes for protein is encoded by DNA sequence. So almost all the relations are undirected. It's just different semantic, how we call it. Like as far as you can kind of like make it kind of directional if you want to kind of force it, but we go with undirected as far as.
But if they're undirected and you're writing on the edge that this one encodes this one and not the other way around, it's like saying that they're directed.
Yeah, but so it's because when I say encodes, so the same way I can say protein is encoded by this DNA sequence. I then kind of put both semantics to reflect both directions of the relation.
Okay, but with DNA and protein sequence, it's quite clear about the connections. But if you have two strings, for
Yeah.
example, two DNA strings,
Yes.
how do you know from looking at the edge which one is a substring of the other?
Well, it's
Without putting a direction on the edge.
it is kind of like it is kind of like represented in database. It is represented in a directional way. Yeah, it is.
Okay.
Conceptually, though, it's undirectional. So it's kind of in the sense that this is like a substring and a superstring the other way, but you pick one direction as far as the actual implementation. That's true.
Okay, but they're undirected because there's no, the edges are of different types.
Yes.
So there's no really.
You have, each two edges, like when you talk about the two edges here, so they're exactly different type, different semantics. Yes. So each one of those edges can have, can go both ways. So again, this goes on. For example, this is this protein manifests itself in this protein structure, or you can just kind of, change kind of the sentence to reflect that the structure is basically a representation of that protein sequence. And the same thing here is so this in this case the edge kind of represents the fact that this protein is involved in interaction, or this or the other way around. This interaction contains that protein.
Okay.
So
But there are some types, at least in this level of edges, which can be thought of as undirected. For example, that this is similar expressed or similar.
yes, there are some that they
Undirected in the nature and some are not.
yeah, I mean, like you can, yeah. Yes, I agree. So the other types of relations that we have here in data are similar. For example, here we have two protein sequences that are similar. Here we have like in this case it was similar based on sequence. Here it could be based on expression data. And we have two proteins that based on mRNA expression data, we can deduce that they are similarly expressed and so on. Here we have. This protein is part of the protein family. It could be an enzyme family, for example. This enzyme family is could be a part of a metabolic pathway, for example. And so enzymes are proteins that catalyze reactions, enzymatic reactions. And then there are many metabolic pathways that consist of multiple enzymatic reactions. Could be in a sequence, could be in a circle. So pathways kind of like those are the cellular programs that we mentioned before. So this is just an example of like a small.
So, just a question. So what are your the numbers? So how many pathways do we have? How many protein families do we have? Just to get an idea on how big.
Right. So you have like tens of millions of DNA sequences. You have like 20 million protein sequences. You have a much smaller number of the other data sets. So you have, as far as structure, you have less than 100,000. As far as interactions, you have also less than 100,000 and so on. So
it decreases like exponentially the number.
I think we have enough data to. But I mean, those are to infer interactions. That requires those are some new technologies that enable you to measure those on a cellular level. It's still much more expensive and involved. So compared to the fast sequencing technologies, it doesn't keep up in the same pace.
No, because maybe you would want to navigate different structure in different ways depending on how many you have for each class.
Sorry again?
No. Go ahead, sorry.
Okay. But it's true. I mean, definitely, it's not like equally kind of distributed. I mean, the majority is DNA sequences and then protein sequences. But the highly kind of the presence of interactions or pathways is highly critical in order to kind of characterize entities. So that provides you with a lot of information about information transfer between different molecules or their the cellular pathway. Gives you the kind of the context as far as the biological procedure that this gene is involved in and so on. You mentioned another main principle of designing this resource is that the keys when we talk about entities and we want to determine which entity we are looking at, so we are not using accession numbers, but rather the physical objects themselves are the keys. So in order to figure out that we are looking at the same entity from different databases, for example, when it comes to protein sequences, when we download different data sets of protein sequences from different resources, so basically we look at the actual protein sequence, and the sequence is identical, so that's, then we decide that we're looking at the same, we're looking at the same entity. There are some exceptions, as always, in biology, there's the rule, there's the exception, so, but in general, this is kind of, this approach helps to eliminate redundancy and aggregates knowledge. And it starts from the individual physical objects and it propagates, this principle propagates to sets of entities. So basically, if you kind of determine, individual kind of, when, for example, we want to determine, that two different interactions from two different databases, actually the same interaction. So it's basically a set identity problem. If you kind of, if you determine the identity, the level of the most basic objects, you can propagate that to the more complex sets in order to determine the identity of the level of interactions. And that also applies when it comes to pathways and so on. It just becomes more and more complex entities that you're dealing with, but the principles are still the same. And just to mention the last principle that I mentioned here is that updates, when we update the resource, it's done under pretty strict consistency criteria. So we don't want to contradict the information. We don't want the information in Biozon to contradict the information from the source databases. At the same time, we don't want to create internal contradictions. So there are all sorts of protocols that are in place to kind of to try and accommodate that, and also to trigger, whenever something changes in one of the source databases that can affect one of the relations or another piece of information, another entity in Biozon. So there are graph triggers that are triggered to check the validity of all those relations or other type of metadata that was computed from the original entity. So this kind of basically, if we're going back to that graph, if we make any change here, we also kind of we reassess, the other relations kind of to make sure they're still valid. So even if the original database that we updated didn't kind of say anything about those relations, but we have to make sure they are still consistent internally.
What happens if you look at two other databases and you find out contradictory data?
If you find out that what?
Contradicting
data?
Yeah.
So we don't try, we don't kind of the approach we took was to kind of create an image of the source databases, and what happens is either we kind of create a new, it could be kind of clear through an example, but the idea is that, at any given point, the data that we have in Biozon doesn't contribute to source databases, so it means that, let's say you had two sequences in two different databases that will merge into the same entity in Biozon at some point. And then one of the databases kind of decided to change its entry and change the amino acid sequence of one of those proteins. So when we update, the information from that database into Biozon, so it's split, exactly, it will split into two different entities. So, and that's a very simple example. There are much more complicated scenarios that we have to address like this. So, now another thing that we also introduced in Biozon is that data is never deleted, so we use timestamps to indicate what is the timeline, the relevant timeline for every piece of information. So you can basically kind of query the database as of any date in time, any date, and then kind of get a picture of the data that we had at any point in time. So what can you do with Biozon? So at this point, I'll teach you the demo. So the first thing you can do with Biozon is view all the information is available for a specific biological entity and all the entities that are linked to it. So this is the interface to Biozon, biozon.org, and this is a very simplified kind of graph which just shows the general entities that we have. This shows some of those with the relations between them. And you can start by, actually before I'll show you the broader context of a specific entity, you can start by just doing a quick search, for example, ubiquitin, which is a protein involved in cellular degradation. This is basically the protein that is used to kind of mark a cell for basically apoptosis, which is a cell that indicated it had to go through the process of being recycled, basically. So this shows you if you kind of submit this query It shows you kind of just here it shows just the top 10 results from each of those data types. It shows proteins that match this pair. It shows DNA nucleic acid sequences that match this search term, the structures, pathways, interactions, and so on. But this is just kind of it's not necessarily the most useful kind of way of creating this data. But let's say you want to start with a specific protein sequence, a specific gene. You want to kind of get all the information associated with that gene. So I'll type just one. Often give an example. So it's a specific, and I'm using here an accession number used by SwissProt. It's one of the databases, one of the common databases. So they use, for example, those kind of accession numbers to refer to accessional names to refer to proteins. Remember, in the very beginning, I mentioned one of the problems with dealing with all this data is that every database has its own set of identifiers. Right? So while in Biozon we strive to kind of deal with that and being able to determine which are the same entities, we also still want to enable researchers to query this data in the way they're used to. If they're used to SwissProt or other databases identifiers, they can still query this data. All those identifiers are still there. So, so this will just take you to, take it to the specific profile page of that specific protein. So I can see the transcription factor. Over here, transcription factor involved in basically kind of controlling which genes are being kind of activated, deactivated at any given point in time. And this is kind of already you have a nice example here of like what it says here. This is the description from one of the databases, SwissProt, but it says here also defined as, and then you have the name of the database and the description of that database. So this is already kind of like a small nice example of like what happens when you integrate information from multiple databases, because some of them have more detailed description, and this is just the definition. There is more information when you look at the whole record, but as far as the title definition, so some use just this very cryptic kind of like, that doesn't mean anything. And this one, for example, it refers to the sum of assembly regulatory protein. And the thing is that this is kind of like aggregating this knowledge together. You already kind of get a more complete picture of what's going on. This is especially useful, for example, one of the databases might have the wrong annotation, not wrong description. So this kind of can help you to figure out that this is the case because all the definitions are pretty consistent. That one of them stands out, is inconsistent with the others, so it might suggest either that this other database is wrong in its description, or it actually might suggest something that you might want to look into. So this is something to leave to the user. So that's kind of answering kind of like second part of your second answer to your question is that there are certain inconsistencies that we just leave. These are kind of semantic or like content kind of like inconsistencies that we don't try to resolve. We leave it up to the user to kind of look at this information and figure it out, because this is it's very it's very difficult to actually kind of figure out eventually what is the correct function. This is actually the subject of of research by many researchers. So, and then you have when you scroll down, you have information. You have of course the protein sequence itself. You have linked to all those descriptive documents from the different databases, containing information about that. I won't follow all the links, but once you start kind of clicking on any one of those, it becomes kind of a random walk often. You jump from one object to another. This is kind of like information about the domain structure of that protein, which is kind of basically the kind of building blocks of proteins. I don't know if anybody mentioned that in previous talks, but proteins, you can think about proteins as sentences, and then you have domains, which are the equivalents of words. Right? So then, like being able to basically partition a protein sequence into its domains, it's a big problem in computational biology, because by being able to delineate the protein into its domains, it's again like being able to understand the semantic meaning of a sentence. First, you have to understand the words. So the same thing here. Now we have those links to related objects. So we have, in this case, we have 3 DNA sequences that are linked to that specific protein sequence. We have 108 interactions associated with that specific protein. We have Unigen cluster depth information that is relevant to genes. Let's just click on, this is, let's click on one of those. For this one, let's click on, sorry. Yeah, so for example, here we have, when I clicked on that third DNA sequence, the information associated with that DNA sequence. So we see that it actually codes for 17 proteins. So this is a longer fragment of DNA that codes for our protein in addition to 17 other, in addition to 16 other proteins. So it's, again, it's kind of like a, as opposed to the two other DNA sequences that were shorter and were just associated with our protein sequence. So this is, and when you click on that one, you can kind of see what other basically genes are, what other proteins are coded by that specific subset of DNA. And you can, here when you click on interactions, you see information about all those different interactions that are mentioned previously. They're very critical to store information or basically transmit information to different molecules, and so this is kind of a very important mechanism of cells to process.
I have a question. I have two questions on that part actually. So are you referencing from citations at all? Like are there abstract, like PubMed, or actual papers that are?
The information, there is, for example, when you click on Speedsport, for example, so usually there is like, where is it, here. We're not using it explicitly stored, but it's another type of information that still has to be utilized. But it is there, it's stored in the database, and all those papers that specifically studied that specific protein sequence, that list.
You can do the reference.
Yeah.
Is there any, I was wondering too, like with the protein interactions, in terms of like the contribution data, is there any weight to, if you have three sources that say these two proteins do interact, and two, is there any way you can?
Yeah, it's a very good question. So actually, when you look at interaction, I don't know if this one is, sometimes, here, for example, it says here, at the top, you see method cross-linking. If this interaction is documented in different databases or in the same database, but documented as, it was determined using several different methods, so you'll have all this information here saying that it was, the methods is cross-linking, like these two hybrid and so on. So that kind of helps you to kind of decide whether this is something that you could strongly believe in or not. So the information is there. And then you have also information about similar protein sequences. So this is something that, for example, here, it's similar to another transcription factor. You can view the alignment and so on. So this is like, that's the similarity data that I was referring to before. It's already computed. This is the kind of metadata that we compute in Biozon and store it in the database and to utilize it kind of further. So that's kind of the first kind of type of query that you could kind of perform in Biozon, just specific accession number, specific identifier. You can just draw about the context of this one. But the real power of this system is actually its ability to perform what we call complex queries, which are actually semantic queries. Meaning, let's say you want to study a specific gene and you want to kind of interfere with the activity of that specific gene. So let's take as an example BRCA1, which is a gene that has been linked to breast cancer. So let's say if you want to kind of target that specific gene, one of the ways you could go about it is kind of try to kind of find other genes that interact with that gene, meaning that they kind of they form a complex with the gene. And then by studying the way those genes interact, or by studying the shapes of those proteins, basically, and exactly how they form the interaction, how they form the connection, you can possibly create other molecules that mimic those mechanisms. For example, you can create a drug that has a similar shape. If you're able to determine exactly the interaction and determine the interacting site, so you can identify those complementary shapes, and you can then possibly design a drug that kind of like is shaped, in a way that it can go inside this kind of groove that exists in order to kind of interact with the other molecule. So this is kind of the motivation behind if you want to kind of interfere with the function of the gene, let's go and collect structures of genes that interact with the gene, understand exactly how they interact, and then possibly can design another molecule. Or we can just interfere with that interaction site. Okay, maybe kind of it doesn't necessarily have to kind of be in the shape or in the complementary shape, but it's enough that it just binds in that interaction site, and then it prevents the other gene from interacting. So in order to collect this information, right, what we let's see how we collect this information from Biozon. So what we want is structures. So we'll start from structures because we want to study the shapes of those genes that interact with BRCA1. So we'll start with structure. You can put all sorts of constraints, for example, resolution and so on, but I'll just continue for now. And we want, so we want those structures to be of proteins, obviously. So we want those structures to be of proteins, and I'll just. So this is the first part. We want structures of proteins that interact with BRCA1. So we just build it very logically, as the way we think about it. So what we want to do next is link those proteins to interactions, because we want structures of proteins that interact. Right. So the next step is interaction. We are changing the focus of the query here to protein, because we want the protein to be the interacting partner here. So we kind of it's kind of, I don't know if you can see it, the color kind of changes kind of slightly from, kind of light pinkish kind of color when we change the focus of the query. Initially it was structure, now it changes to kind of protein. I don't know if you can see that. But anyway, now we'll click on interaction, and here it's indented to reflect also the structure of the query. Okay, so what we are searching, again, there is a summary of what we're searching for so far. We're searching for structure of a protein. That protein is involved in interaction. We want the other side of the interaction to be the BRCA1 gene. Okay, so we're going to continue with building this query, and we're going to change the focus of that. We're basically building a graph query. Okay, so we kind of change it, we're deciding which node is the node we're going to focus on now, and which edge we're going to introduce now. Right, so we're going to change the focus to interaction. We're going to kind of now introduce the other end of the interaction, which is again a protein. Okay, so then we're searching for a structure of a protein that is involved in interaction. This interaction involves another protein. This other protein, now we're going to put the gene name BRCA1. Okay, so this is, again, we're just going to, so this, take a bit, but basically, it's doing exactly what we asked. We just kind of build the query, kind of following this kind of logical steps that,
Do you have any plans, now they're building these atlases that show gene expression, like actual profiles where people have done?
expression data?
In terms of visually, where people show an atlas of where different transcriptions, with Biozon, are you planning to incorporate any of those?
If there are some graphical actually displays of certain aspects of the information, definitely, yes. I kind of skip, when it comes to gene ontology, for example, we have a graphical display of that. When it comes to interaction maps, we have some kind of ways that come to kind of display graphically those interactions, kind of, because all the connected components, it's a specific connected component consists of all the different interactions and so on. So this is kind of one example that we, yes, it just there's so many types of, there's massive amount of information out there, different types of, so many applications you could use. So yeah, it's just, I guess you can call it the prototype system, and there's still a long way to go. So this is, these are basically the answers to our query. All these are structures of genes that interact with BRCA1. When you click on each one of them, you can, each one of those is a protein structure. You can download it and so on. So I don't want to kind of go over time, so I'll just, I want to kind of continue with this example. So the next kind of type of query I want to demonstrate, we can kind of deal with Biozon, is what we call fuzzy query. So remember we talked about metadata. I mentioned that in Biozon we introduce all sorts of metadata, including similarity relationships between entities. So we take the raw data and we compute, for example, between protein sequences. We compare all protein sequences to all protein sequences to determine which sequences are significantly similar to each other. If they're similar, often the idea is that sequences are statistically, if they're similar, the similarity is statistically significant, you can often infer homology, which means that they probably evolved from the same ancestor protein. Usually they have the same or similar biological functions. And this similarity relationship is fundamental to biological inference, because often, when the amount of data that is produced is such, there is no way that you can actually run experiments on all those entities. So most entities these days are actually characterized by characterized computation. You compare, for example, you take a protein sequence, you compare it to all the other protein sequences, and you try to extrapolate the properties of the, you look at the most similar proteins and you try to extrapolate their properties to your protein. So this is the most common way of inference. Of course, it's subject to, you always have to kind of like remember that this is, even if it's similar, it might have, the variation is such that it might affect the function in a certain unknown way. And if you really want to understand what this protein does, the new one that you just compare to all the others, you have to study it in the lab. It's not enough, but this is a major step towards at least providing the first clues. So by computing all the similarity data and storing all the similarity data in Biozon, what we are able to do is actually propagate knowledge from well-studied entities to unknown entities. And so the idea is that, let's say you have, and I'll demonstrate this kind of simple query. Let's say you start by just looking for proteins that are involved in autoimmune disease. So I'll put here autoimmune, okay, so this gives all sorts of proteins where the definition has the word autoimmune in it. Okay. Now, what I'll do next, I'll revise the query, and I'll add at the bottom here. There is, you see those options here, and I'll say here use. Check here, use results of last alignment to include similar proteins in the query. Which means that now we're going to search for all proteins that have the keyword autoimmune in their definition somewhere, and all the proteins that are similar to those proteins. Okay? So, and the idea is that this is something that if they're similar, the similarity is significant, so they might be very relevant to my query. And this is the major shift compared to, for example, when you do web search. When you do web search, the only web pages that will come will be only those that have actually the keyword. But here, what we can do is actually we can bring forward also other pages or entities that otherwise we would never have looked at. And this is so the way the information is displayed. Let me change that to display. So there is also such that first all the exact matches are displayed. There are links to all those different proteins, and then at some point you see those green boxes. Those are fuzzy matches. So each one of those is a match that came because there was a similarity with another protein that had the word autoimmune somewhere in its definition. Now the cool thing about so this is kind of all those entities might be kind of like interesting, but let's put another component to the query. Well, let's add ranking. So the ranking kind of concept is something that early on we realized, well, we have this kind of very large graph, similar to the web. The same way you kind of utilize the web structure in order to determine which entities, which web pages are more relevant to your query. We ask ourselves, can we do something similar when it comes to biological entities? Can we report the results instead of an arbitrary ordering, which is what all the databases are doing? Basically, they're just returning the results when you kind of format, when you submit a query. There is no kind of real sense of, there is no reason why one entity would be kind of like before the others. It could be kind of, some databases just use the date, the entry was included, some kind of like, what? But, well, they don't use pager, but that's what Biozon did, what we implemented actually kind of what, we implemented something similar to what Google are doing, which is kind of, well, it used to be pager. Google doesn't do pager anymore. But we played with different models that are used in the web search community. PageRank is only one of them. There's hubs and authorities. There's agent layer centrality. All those methods, what they do, all those models, they look at the connectivity matrix of the graph. And there are different ways to define the connectivity matrix. So basically, they analyze the spectral properties of this graph. And based on that spectral properties, they kind of come up with a certain ranking. The idea is that the prominence of every node in the graph is a function. Is a function of the other nodes that are connected to it. And different models basically define this kind of connectivity, define the dependency between nodes in different ways. So that's kind of what differentiates kind of the different models. So I'm not going to go into all that. Just to kind of like give you an idea about what's. And actually, we tested all these models, and we ended kind of like concluding that page or variation of the page rank actually worked better than all the other models when it comes to ranking biological entities. So right now the results I introduced kind of, as I said, initially it's just the exact matches, and then the fuzzy matches. But what I'm going to do next, I'm going to rank the results, which means that we're going to use those prominence values that were computed based on the complete graph structure, okay?
You had a photo?
Yeah, this is basically kind of asking, like, was that useful? We're asking the user, can you tell us whether this ranking was useful to you? Because we're trying to gather information on whether the ranking, because there's so the way we kind of like, we figure, how do you rank the results of, biological parameters? But there are, it could be the rarest kind of variant is the one you're interested in. Or it could be kind of like, could be the one that is like from a specific organism. But in general, on average, the method we kind of, the approach we took was that, well, in the absence of any other kind of like preference, what we will follow is simply the more information you have, the better. If you have, for example, if you're looking for a specific gene in human, right, but you haven't, this gene hasn't been studied extensively, you don't know much about its function, but there is a model of this gene that in mouse, for example, has been studied extensively. Many of its interactions are documented, cell type and so on. So bringing forth this kind of gene, from mouse, because it's very well documented, could really help you with analyzing your entity. And this is, so basically the motivation is, again, if the more information you have associated with your entity through those connections to other entities, it might be more interesting to you. It might kind of like bring more, we call those hubs of knowledge. It might bring more knowledge that could be useful to you. And what happens here when we rank the results based on those Provenance values, we see here that, for example, the first match is actually a fuzzy match. Right? It's a kind of it doesn't have the word autoimmune in it, but it might be highly relevant. You can kind of, by clicking on this match, you can kind of further explore, what is going on, why is that protein, relevant to your query term and so on. So, I think I'll stop at this point. This is just to conclude. And there are more things you can do with Biozon, but I hope I give you an idea of what kind of what is the power of such a system once you integrate all the information into a single kind of infrastructure. But it is a grand challenge starting with the fact that you have to deal with massive data sets, dealing with those all those inconsistencies, different formats. You have to kind of when performing complex queries, those optimization issues that everywhere you look, there is a different kind of problem here. But this shouldn't kind of discourage us from kind of approaching this problem, because once you build such a resource, it's really critical for analysis of complex and interrelated entities, and it's highly relevant to new directions in medical research. You've probably heard the term personal genomics. This is the direction that medicine is going towards. The idea is that instead of going to the doctor and having just blood tests, you can hopefully in a couple of years you'll be able to submit a sample of saliva or something like that that would kind of be used to kind of determine will be sent to determine your DNA sequence. Then being able to kind of upload this DNA sequence to a system like that and and connect it to all the different entities, right? And giving kind of basically the context for this context that will help disease diagnosis and treatment. To fine-tune kind of treatment to a specific genetic make of each individual, as opposed to what is going on today.
And to anxiety.
That's what?
And to anxiety.
Actually, one of those. It's just there's so many. I don't know if you have. Hopefully, you didn't have to go through an experience, but you did. It did happen to you. You know what I'm talking about. You go to the doctor and try to describe what's your symptoms, and they try to kind of basically kind of take what you're describing and pick one of the specific diseases or disorders that they're kind of familiar with. And there are many of them. This is kind of deciding on the right one. This is kind of often it's kind of like a trial and error. You go and it's not the first time, and they try a certain kind of drug, to kind of stuff, but it doesn't necessarily work. So hopefully this kind of could be saved once we kind of with this kind of personal genomics. This is something that could really kind of change the way things are being done. But all this cannot be done without really a community kind of effort involved, because when we're dealing with massive data sets produced by different research groups, different formats, different applications, different kind of tools that you want to do this. It's there is no single kind of group or team that can do that. So we're building this resource, hoping that also this would kind of encourage others to be involved. And one of the one of the engines that we kind of like hope would kind of make that possible is that we also enable researchers to deposit their data in Biozon. So it's kind of like instead of going and building their own database, spending resources, and they might not have those resources or the expertise, they can just upload their data into Biozon, immediately link it to the wealth of information out there without going through all this kind of process. So this is something that we hope will really change the way, the momentum of the way things are being built. So